Hi, my name is Ayesha Tariq and I am a PhD candidate in Dr. B. and Kiska's lab at Kent State University. In this presentation, I will be talking about the effect of ADAR editing on microRNA target binding in autism spectrum disorder. Autism spectrum disorder is a neurodevelopmental disorder associated with the genetic and phenotypic heterogeneity. ASD is commonly characterized by symptoms such as repetitive behavior, ADHD, and social and behavioral deficits. Common risk factors, genetic risk factors associated with ASD are chromosomal abnormalities, rare copy number variants, and point mutations. However, recent reports from human postmortem brain samples from autistic individuals have shown that not only chromosomal abnormalities but also transcriptome wide dysregulations in terms of differential gene expression and alternative splicing are also associated with ASD. In the light of that, a very important mechanism that regulates transcriptome in terms of adding diversity to transcriptome is the editing of double-stranded RNA by adenosine deaminases acting on RNA or ADARs. ADARs can bind to an adenosine in a double-stranded RNA substrate and deaminate the adenosine into inosine, which will then be read as a guanosine by the translational machinery. Similarly, microRNAs are another important regulator of gene expression. MicroRNAs are small non-coding RNAs that can bind to a messenger RNA and stop its translation. Studies have reported differential expression of microRNAs and widespread ADAR editing in autistic individuals, but no studies, if any, have looked at the correlation between ADAR editing and microRNA binding to their gene targets. Therefore, to address this gap, we analyzed a whole brain transcriptome data from Kim et al. This data comes from ARID1B heterozygous mice, which serve as autistic mice model and will be referred to as HT mice in this study, and wild type mice, which will be referred to as WT mice in this study. Half of these studies mice were treated with the drug, but since the goal of this study was not to look at the effect of drug on differential editing or microRNA target binding, but for a direct comparison, we compared the drug treated group with the drug treated group only, and similarly we did the same thing for vehicle treated group and compared vehicle treated mice with vehicle treated mice only. To identify ADAR edited targets, we identified A to G or T to C variations using AID automated pipeline, and then those identified targets were analyzed for following consideration. Number one, change in the levels of editing. Number two is the differential microRNA binding to those differentially edited genes, and finally the gene list was analyzed for its involvement in KEGG pathways. Following an analysis, it can be seen in figure 2 that the expression of ADAR1 and ADAR2 has increased in HT mice as compared to WT mice. To see that if this change in expression has translated into any editing changes, we looked at differential editing broadly in terms of shared edited targets in WT and HT groups, and narrowly in terms of uniquely edited targets only in HT group. For shared edited targets, to see if there is a change in editing, we calculated odd ratios for those targets with a 95% confidence interval limit, and any odd ratio value greater than 1 indicates a high risk of editing in HT group. After the identification of differentially edited targets, we extracted FASTA sequences for those respective genes, 100 base pairs flanking the edited position, and then those FASTA sequences were analyzed for their binding with a set of microRNAs extracted from miR-based database for mouse mRNA and mouse miRNA, and RNA22v2 tool was used to see the binding dynamics of microRNA with those differentially edited genes. Results from differential editing and microRNA target binding analysis show that there is a difference in the number of microRNAs binding to the gene, in the type of microRNAs binding to the gene, and the strength of microRNA binding to that gene, which is highlighted as changing holding energy values in this study in edited versus unedited state. We further compared our list of differentially edited genes that had one or more microRNA binding targets with Safari gene list for autism-associated risk genes. There's only one gene from our list that overlapped with Safari gene list, which is STXBP1. STXBP1 facilitates the release of neurotransmitter in synapse, and deficiencies of STXBP1 in mice are related with cognitive impairment, anxiety, and social and behavioral deficits. Conclusively, our analysis show that there is a difference in ADAR editing patterns between autistic mice models and wild type mice, and those differences does affect the binding of microRNAs to differentially edited gene targets. Future studies should look at the experimental validation of these identified targets and also focus on their downstream consequences on protein translation. Thank you.